What we're trying to do is to try to actually help human health. And so it's become a lot easier and over the years to actually be much closer to making that a reality. It used to take a long time for the discoveries in the lab to actually be transformed into something useful. But nowadays, with particularly with the increases in the technology and what we can actually do, it's a much faster process to think how the discoveries in the lab might actually convert to things that are useful, such as drugs or vaccines for preventing human disease. What my lab has been interested in is how microbial pathogens, that's things like viruses and bacteria, are recognized by our immune system. So when we get a viral infection, what happens? How does our immune system actually deal with those? How does it clear them? How is it responsible for controlling, for example, viruses? So over the years, we've worked on influenza virus, we've worked on HIV, we've worked on hepatitis virus, and a number of different other pathogens. My lab consists of around 25 people. It's composed of graduate students, postdoctoral fellows, research technicians, and administrative personnel. So we've been working on the HIV trimer for well over 10 years. This started as a collaboration with John Moore's lab at Cornell. And so one of the difficulties is that this trimer is quite unstable. And so one had to actually work on trying to get some form of the trimer that we could actually use for structural studies. So about 4 versions later, over a period of the last 12 years, working with John's lab at Cornell, we actually got a trimer that could be crystallized and we could determine its structure. At the same time, about a couple of years ago, we started working with Andrew Ward's lab here on electron microscopy. And so these trimers that were useful for X-ray work were also suitable for electron microscopy. So that enabled our lab and Andrew Ward's lab to be able to get structures of the trimer around the same time. And so this was a tremendous breakthrough. This is something that we've been looking forward to for many, many years. And it was really sort of a synthesis of these 3 individual labs that enabled this work to come to fruition. In order to try to figure out how to get a vaccine, a lot of the focus has been on structure-based vaccine design. And for structure-based vaccine design, you have to have a structure. We'd never had a high-resolution view of the details of what an HIV trimer looked like until now. So we now can use that information for looking at where antibodies bind, how we might stabilize this trimer, how might we use this trimer as an immunogen along the path towards getting a vaccine. What we've been trying to work towards is a universal flu vaccine. So instead of having to take a vaccine every single year, it would be really nice if we could get a vaccine for life, or certainly one for 5 or 10 years. The universal vaccine is probably still several years away, but we know now that it's likely to be possible. For me, a typical day is it's always a very busy day. During term time, it's grad class in the mornings, Monday, Wednesday, Friday. There's usually a series of meetings. There's a lot involved with the grad program. Is then trying to find out what things that are going on in my lab, and then trying to sort of figure out what we're actually going to do for the next few weeks. When I first came 30 years ago, TSRI was largely known for immunology, and so this was really the start of an expansion. Richard Lerner got some funding from Johnson & Johnson to start molecular biology, and that really started off the sort of the second phase of TSRI. Shortly after molecular biology, there was cell biology, chemistry came, and so there's really been huge advances over the years in the number of different areas that we actually cover here in biomedical research at the Scripps Research Institute. Another important development was the graduate program, and that started in 1989. And so for the first time, we could actually take in our own students and give degrees to them. We have, I think, historically, from the first time I came here, we actually worked collaboratively with a number of different labs. I think that was very unusual at the time, but actually we realized that if we combine our strengths, we could actually move along a lot faster. It really enables us to push the frontiers in the areas that we're working in.